So it's a privilege to introduce Dr. John Svendsen for this special seminar here. I actually met John through, I guess, a very new way of doing things, which was 10 months ago on LinkedIn when I posted something in a fish ecology group, which was, I'm interested in doing fish avoidance work. Does anybody have any ideas of what flumes and what kinds of devices I can use? John messaged me saying that, you know, I have these 2 flumes in Denmark that are sitting on the shelf. If you want, let's collaborate. I'll ship them over to you. Long story short, they're in Southampton and they're going to be used this summer. John got his PhD from the University of Copenhagen in 2008, and his dissertation looked at the migratory behavior of salmons in fresh water in relation to physical barriers. He's done multiple postdocs. This work that he's presenting is from the work he did at the Technical University of Denmark. He's also worked as a postdoc at the university in Minneapolis, where he was tasked with trying to create carp barriers for carp. He's also worked on some top secret work at the DFO in Canada, which he hasn't been able to elaborate with me about. And he is currently going to be going to the University of Porto to develop new ways to look at deep ocean fish metabolism in relationship to hypoxia. So thank you very much, and I'll turn it over to you, John.
All right, thank you very much, Konstantine. I will get going with my talk about interindividual variation in aerobic and anaerobic metabolism in swimming fish, and we're going to be looking at effects of gait transitions, pectoral fin use, and reproductive traits. And as Konstantin was just telling you, that this is stuff that I've been doing at the Technical University of Denmark. So this is my presentation outline. We will start out by having a look at the importance of metabolic rate and locomotor performance. And then we'll take a look at aerobic and anaerobic metabolic rates and how to measure them. And that is usually done by having fish in a respirometer, and you can use intermittent flow respirometry. Then we'll have a look at the gait transitions in terrestrial and aquatic locomotion. And we'll continue by having a look at pectoral fin use and swimming stability, and reproductive allocation and pregnancy stages, and then our hypothesis and predictions, followed by the methods, results, and the conclusions. Right, I study locomotion, and I do that because it's important for a lot of aspects of fish. For example, the reproductive migration, foraging, and territorial defense. As we can see the brown trout here, and the eel is known to migrate from Europe all the way to the Sargasso Sea. And juvenile dispersal and colonization and invasive species here as the Asian carp. And all these behaviors are energetically expensive, and that's a characteristic of the aquatic environment. And that brings me on to the metabolic rate. Recent studies have found that there is substantial interindividual variation in aerobic metabolic rates, so that one fish can have a standard metabolic rate or a basal metabolic rate of about 100, and then a fish which is basically the same, it's the same species, yet it has the standard metabolic rate of about 300. And this should have substantial consequences for the individual, because throughout its life it's going to be struggling with this high standard metabolic rate. So what is the advantage? Or yeah?
Is this already like accounted for the body size and the
Yes.
Okay.
Yes, that is corrected for body size. Yes.
So a variation of threefold.
Yes, up to 3. In some species it's up to 3. In some species it's 50%. And this has causes and has consequences, and they are uncertain. But there is an emerging pattern suggesting that these metabolic differences are linked to behavior or life histories of the fish within the species. And for example, Killen et al. In 2011, they showed that schooling fish, that the leading fish, they have a better aerobic capacity than fish in the rear end of the school. So that metabolic capacities can explain some of the variation or some of the reasons why the fish are positioned various places within the school. With that, I'll move on to the basics about aerobic metabolism. And as I'm sure all of you know, that it's aerobic metabolism involves converting nutrients such as glycose, amino acids, and fatty acids into ATP, and that provides energy for the animal. And aerobic metabolism is fairly efficient. It provides 38 ATP molecules per oxidized glycose molecule, which is in contrast to anaerobic metabolism, and that works basically without consumption of oxygen, and that is much less efficient. You only get 2 ATP molecules per oxidized glycose molecule. And it also leads to accumulation of lactate, and various sources of energy in the animal are depleted when the animal is relying on anaerobic metabolism. And so that means that after the anaerobic metabolism, storages of glycogen, creatine, phosphate, and ATP, they will have to be regenerated, and also the lactate should be cleared out of the animal in order to let the animal perform after the anaerobic metabolism. So how is aerobic metabolism measured in fish? It's usually done in swimming flumes like this one. Here we have an eel and we have a mackerel. And these recordings were made by John Svendsen. The method that's typically used is the critical swimming speed here, and that involves having a fish in a flume like this, and then over time, then the swimming speed is increased stepwise all the way up to the level at which the fish can no longer swim. And then at each of these steps, for example this one, then the oxygen consumption rate can be measured using intermittent flow respirometry, and that is done by having this chamber sealed, and then the respiratory cycle starts by flushing the chamber, and that brings up the oxygen in the water. And then there's a wait phase in which the chamber is closed. And then as soon as you get a linear decline because of the metabolic rate of the fish, then you can fit a line to this, and the slope of this line will basically give you an indication or a measure of the metabolic rate of the fish. And then you flush the tank again, and then the oxygen content of the water comes back to 100% saturation, and then you can increase your speed measure, and that way you get one metabolic rate at each of these swimming speeds. From these measurements here, we have Ohlberger in 2006 data on common carp, and from those measurements you can get the swimming speed and you can get the oxygen consumption rate. And here fish with different body mass are measured as they swim faster and faster, and then the aerobic metabolic rate is measured as I've just described. And using these lines, then the lines can be extrapolated to zero swimming activity, which is then a measure of the standard metabolic rate of the fish. Now, as we've just seen, this involves instantaneous oxygen consumption rates as the fish are swimming. But there is the possibility that you have anaerobic metabolism going on at the same time. So the cost of swimming, if we take anaerobic metabolism into account, could actually be higher as indicated by these red lines. So that would be the true cost of swimming when the anaerobic metabolism is taken into account. So how can we measure anaerobic metabolic cost of swimming in fish. As shown here, which is data from Lee et al. 2003, it can be done in a similar fashion. It just involves a slightly different protocol. Again, just like with Ohlberger's et al. 2006 data, we have the swimming speed on the x-axis, and then we have the metabolic rate on the y-axis. And we'll swim the fish, increase the speed all the way up to the critical swimming speed. And then as the speed goes up, so does the metabolic rate. And typically the first speed down here is typically about 0.5 body lengths per second. And that corresponds to this metabolic rate. Now, after the critical swimming speed, then instead of taking the fish out of the tank, you can keep the fish in the tank, and then you can bring the swimming speed back to the 0.5 body lengths per second. And then if all the activity that the fish, or all the swimming activity that the fish was doing, if that was perfectly aerobic, then we would predict that the metabolic rate would return to this level when the swimming speed is returned to this level too. Now that's not always the case. Instead, it may take a while for the metabolic rate to go down, and that constitutes the anaerobic cost of the swimming. And then by integrating this curve, we can get a measure of how much oxygen the fish needed in order to recover from this swimming, which is then an additional measure of the swimming cost. And it's usually termed the excess post-exercise oxygen consumption epoch. And in this case, it was about 87 milligrams oxygen. And then what Lee et al. Did is that they added it to the swimming speed so that they were trying to estimate the total cost of swimming, which then went all the way down to 60% of the Euclid value, and then this would be the actual swimming cost, accounting for both the aerobic and the anaerobic component. What they didn't do is that they didn't verify that the anaerobic metabolism started already here. It could equally well have started at a much higher speed and then just resulted in a higher cost of swimming, starting from a higher swimming speed. So how can we use these data to describe swimming cost? We can convert them from the data showing the swimming speed and the metabolic rate. We can derive the total cost of transport. So now it's not it's a metabolic it's oxygen consumption per kilometer in this case. And then we can see that goes down as the speed goes up, and then it starts increasing again. Now you may wonder, why is it so expensive to swim at a slow speed? And the reason is that this is the total cost of transport, so that includes the standard metabolic rate. So that means that if the fish is swimming really slowly, then per kilometer or per meter, then the standard metabolic rate kicks in and has a huge influence. But again, if this is based on the aerobic metabolic rate, then it could be that the true cost of transport is actually a lot higher when we account for the anaerobic metabolism as well. Another way of quantifying swimming cost is by having, now we have the data on a logarithmic scale, and then we have the swimming speed and the metabolic rate log transformed basically, and then we get a linear line here, and then the slope of this line is also an expression of the swimming cost. And like we did with Ohlberger's data, then we can extrapolate that all the way down to here, and then we can get a measure of the standard metabolic rate. Now,
is this just because of the log scale that it seems like not fitting those?
No, that is because I've just put the line there because there's more line I'm going to add a few more lines in a minute.
Sorry.
Another line is this one,
Yeah.
and this line has the slope here is the same, so the swimming cost is the same, but the standard metabolic rate differs. So, and there are various options. Obviously, it could also be that the swimming costs were less, so the slope was less, but the standard metabolic rate was a lot higher. So I'll move on to the concept of gait and gait use, and I think that the best way to explain that is by showing this almost classic data on horse. And we have the cost of transport, and in this case it's the energy consumed per meter. And then we have walking, trotting, and galloping as the speed is increasing. And these data are, nuts in 2011, they were summarizing these measurements in a graph, and that's where I have these data from. And we have the horse, when it's walking, then as the speed goes up, the cost of transport goes down, and then it starts coming up. And as the cost of transport goes up, the horse changing into trotting, and then the cost of transport goes down again and comes up. And as the cost of transport reaches this level, then the horse will change into galloping. And so that's a way for the horse to control the cost of transport using these different gaits. Similar data have been collected on ostrich and kangaroo, and it also shows that there is an increase when the animals are walking, and when they change to grounded to aerial or hopping, then the cost of transport goes down, and from there it's linear, straight. Sorry?
And then the ostrich becomes a kangaroo.
And then the ostrich become a kangaroo. That's right.
And slams.
Yeah.
So are there gaits in fish? Yes, there are. Here we have a parrotfish recorded by John Flink Svendsen, and it's swimming.
That's yeah. How do you say that?
Triggerfish.
Should be passive trigger. Yeah.
Triggerfish, sorry about that. Triggerfish, and it's swimming against a current going this way, and there is a slow swimming speed, and we can see that the pectoral fins are being used, and the fin here is being used, but the caudal fin is not being used. That's not until it's swimming fast. Then it will start using the tail for propulsion. So that's a gait transition similar to a horse, changing from walking to trotting, for example. And we also see that at this speed, the pectoral fins are not being used. It is basically just the whole caudal region that's being used for propulsion. There is another gait transition in fish, which is changing from fast steady to unsteady or burst assisted swimming. And that involves a change from it usually involves the change from the red to the white fibers. And they differ quite a lot, those two types of fibers, in the sense that the red fibers, they're slow and they're aerobic, and the white fibers, they're fast and anaerobic. So that means that when the fish is changing from fast steady to fast unsteady swimming, well, then it will also transition from aerobic muscle to anaerobic muscles. And as we have just seen, the efficiency of these muscles are quite different. So that would predict that the swimming cost might also be different when the animal is changing from steady to unsteady swimming. Another thing which is not really related to gait transition, but is related more to stability, is maneuvering and steady swimming. And Drucker and Lauder in 2003, they investigated these things in rainbow trout, and they found that the rainbow trout were using the pectoral fins for maneuvering, and then when they were steadily swimming, then the pectoral fins were tucked against the body. However, there are cases where animals will, or fish will, combine both the caudal fin and the pectoral fins. McLaughlin and Noakes in 1998, they investigated that in brook trout, and they found that the pectoral fins, even though that these fish were using the caudal fin for swimming, they would use the pectoral fins at the same time. And there is another example, a recording by John Frank Svendsen, and here we can see that the sea bass is using the pectoral fin, even if it's the propulsion is by the tail. So the pectoral fin is most likely used for stability purposes, but at the same time, it probably could increase the drag and therefore increase the swimming cost. Another thing that could affect the swimming energetics and cost and performance is reproductive investment. And here we have the shorthorn sculpin. And this fish is not gravid in contrast to this one. And James and Johnston in 1998, they investigated the escape response speed of these fish, and they found that when they were gravid, then the escape response speed goes down. So with that, I'd like to move on to the hypothesis of this talk. And we were hypothesizing that similar to terrestrial locomotion, just like we saw the horses, then gait transitions in fish should reduce the swimming cost. And we were looking at the pectoral fin to caudal fin transition and the steady swimming to unsteady swimming. And as we have just seen, the transition from steady to unsteady involves a shift from aerobic to anaerobic muscles. So therefore, that and this involves a change from the pectoral muscles to the caudal fin. So all these changes are likely to influence the swimming cost, and we hypothesized that they should reduce the swimming cost, which would be similar to the terrestrial locomotion. The second hypothesis was that extending the pectoral fins when swimming with the body caudal fin should increase the swimming fish. Here we have again the rainbow trout swimming now steadily without the pectoral fin extended, and then we have the same fish with the pectoral fin extended, and that should then increase the swimming cost. So our predictions was that this fish, which is swimming with the tail without the pectoral fin, should have a line looking like this, and the slope would be this, and it would meet the maximum metabolic rate here, and that would correspond to a critical swimming speed of about 82 centimeters per second. And then the fish swimming with its pectoral fins, it should have a higher swimming cost, so the line here would be steeper, but it would not affect the standard metabolic rate, because when the fish is resting, then it doesn't really matter whether when it's swimming, it is using the pectoral fin. In terms of the pregnancy, we were hypothesizing that the swimming cost should go up, and also the standard metabolic rate should go up. So here we have the data, well, not data, but a prediction from the not pregnant fish, and if we compare that to the pregnant fish, then both the standard metabolic rate would be higher, and the slope, and therefore the swimming cost, would also be higher. So we tested these, the gait transition hypothesis. We tested that in the striped surf perch, in Biotrocha lateralis, and we did swimming respirometry on those, as I've just described, and we were swimming them at 0.5, 0.8, 1.1, 1.4, and 1.9 and 2.3 body lengths per second. And we evaluated the epoch or the anaerobic cost after 1.4 body lengths per second, and at that speed they're just using the pectoral fins, and at 1.9 body lengths the tail is being used as well, and then at 2.3 body lengths they start bursting back and forth. So that's on a transition from steady to unsteady swimming. So at each speed we measure the pectoral fin frequency, the caudal fin beat frequency, and the frequency of bursts. This is the swimming respirometer. We have the motor over here, which is powering a circular current, and the fish is swimming against that. There is a mirror at a 45-degree above, so that we simultaneously get a lateral recording and a dorsal recording. And then the fish is swimming against the current going this way, and there's an oxygen sensor here, so that we can measure the oxygen consumption rate at each speed. With that, I'll just show you the videos. Here we have a fish that is swimming only, this is at 1.4 body lengths per second, and it's swimming only with the pectoral fins, and we have the dorsal recording as well up here. And then when the fish is using the tail, it looks like this. So it's a combined way of swimming where the tail is used intermittently and the pectoral fins are also used. And then the transition to the burst assisted is here, and we have the fish bursting and back and forth inside the tank, and then we quantified those bursts. All right, so here we have the data. So we swam the fish all the way up to, so this is basically the baseline metabolic rate at 0.5 body lengths per second, and then we swam the fish up to 1.4 body lengths per second, and then we found that there was no difference. So there was no evidence of epoch. This metabolic rate was not statistically significant from this metabolic rate. So there was no evidence of epoch or anaerobic metabolism, and the same thing was the case after 1.9 body lengths per second. And at this time now the fish is using the tail, but there's no anaerobic component, in contrast to when the fish is swimming 2.3 body lengths per second. Then when we returned the swimming speed to the 0.5 body lengths per second, then the metabolic rate remained high, significantly so, higher than this baseline level, and it took up to 3 or 4 hours for the metabolic rate of the fish to go back to the baseline level here. And then we used this equation in the individual fish to quantify the area under the curve here. So we fitted this line, had it go all the way back to the baseline level, and then we integrated the area under the curve, and then we had a measure of the oxygen or the anaerobic metabolic rate here described as the epoch. So here's the data. We were hypothesizing that the metabolic rate should go down after gait transitions. That is not the case in fish. Here we have the fish swimming at 1.4 body lengths per second. It's using the pectoral fins, and then it transitioned to using also the tail, but there's no difference in the metabolic rate between these two. So this fish does not, or these fish do not change the cost of transport, or the cost of transport does not go down as a consequence of gait transition from using pectoral fins to using a combination of the pectoral fins and the tail. And then here, the fish is starting to use the burst and coast, well, not really, but burst assisted swimming. And then we see an increase in the aerobic metabolic rate. So even if we're only considering the aerobic component, there's no evidence that the cost of transport goes down after the gait transition to burst assisted swimming. And if we combine the aerobic and the anaerobic then there's absolutely no no decrease in the cost of transport. In fact, it increases quite dramatically. A thing that I like to point out here, and which is the focus of this talk, is that there is much more variation in the metabolic rate when we include the anaerobic here as compared to the variation in the measurements when we only included the aerobic measurements. If we take a look at these data, so that now all the fish are swimming at 2.3 body lengths per second, and we have the number of bursts, and then we have the cost of transport. Then we see that the cost of transport is ranging from about 0.13 up to about 0.2 milligrams of oxygen per kilogram per meter. So that's the measure of the aerobic variation, and there's no correlation between the number of bursts and the aerobic metabolic rate. But if we include the anaerobic or add the anaerobic metabolic rate on top of the aerobic, then we see that there is a lot more variation in the measurements. Now the total cost of transport is ranging from about 0.15 up to about 0.35, so it's more than double. So depending, and these are all individual fish, so even if all these individual fish are swimming at the same speed, they may have quite different swimming costs, and that is this increased variation is because of differences in the anaerobic metabolic rate in these fish. The next part of this talk involves swimming respirometry on female Trinidadian guppy. And we were interested in testing if the pectoral fin use and burst activity affects the swimming cost. And we were also interested in testing if the reproductive allocation and pregnancy stages of these female guppies, whether that affects the standard metabolic rate and the swimming cost. So we captured guppies in the wild with these small butterfly nets, and we brought them to a lab, and then we tested them in a little tiny Alaska type swimming respirometer, and we were testing guppies, but this is a zebrafish in here, and so this Alaska type swimming respirometer works this way, that there is a propeller here that drives a circular current that goes around, and the total volume is 0.17 liter, so it's pretty small. And then the fish is swimming against this current, and then as the speed is increased, then we can measure the metabolic rate of the fish. And these pictures were downloaded from Loadigo System's webpage. And in this way, we tested 18 guppies. And here is another view of the respirometer. We were having this tiny little fiber optic oxygen sensor and the motor outside, which is basically driving the current inside. And then we increased the swimming speed 0.5 body lengths per second, and we monitored the pectoral fin use and the burst activity. We did that all the way up to the critical swimming speed. Then we measured the epoch by returning the swimming speed to the speed at which we started the test. And then afterwards we dissected the fish in order to quantify the reproductive allocation and the pregnancy stage. And the reproductive allocation was quantified as the mass of the embryos divided by the total body mass, so it's a fraction of the body allocated to reproduction. Here we have the results for the 18 different guppies, and we have the blue lines, which shows the pectoral fin use, and we have the burst assisted swimming, which is indicated with the red line, and it's all in a percentage of time. And as we can see that there are burst swimming is not recruited or used until the fish is reaching high speeds. It's not all fish that are using burst. For example, here there's a fish that doesn't burst. Another characteristic of this data is that there is a lot of variation in the pectoral fin use. Here, in this guppy number 9, the pectoral fin use just went down at a fairly low swimming speed, and then it didn't use the pectoral fins at all. Whereas other fish, for example, guppy number 2, continued using the pectoral fins almost all the way up to the critical swimming speed. And other guppies, like guppy number 3 or guppy number 14, were using the pectoral fins at all the speeds, all the tested speeds. If we take a look at the same, or the metabolic rate data for the very same fish, then we have the 18 guppies, and now we have the log oxygen consumption rate, and we have the swimming speed. Then we have the metabolic rate, and we fit the lines as I was talking about before. And we were fitting the lines to the data without the burst, so that we were only looking at the steady swimming gait, so the gait without any burst activity, and therefore presumably without any influence of the white or anaerobic muscle. And then we can see that the aerobic the metabolic rate increases during the steady swimming, and then as the fish is transitioning to unsteady swimming, then the metabolic rate, even the aerobic metabolic rate, is increasing, and that is a characteristic of pretty much all the fish that the red data points are much higher than the gray data points. And then we fitted the lines, and then we had an expression of the swimming cost in all these individual 18 fish. Now I'm sure that you are all thinking, now John, what about the epoch, the anaerobic component? Why did you include that in that graph? And I really wanted to include that data in the graph, but I couldn't because I could not calculate it. Here we have, so this is after the UCRIT test, so this is basically when the fish is swimming just before it is exhausted. It has a metabolic rate of about 1,250 milligrams oxygen per hour per kilogram, and then the swimming speed is returned to the starting level, and then if there was no anaerobic metabolism, then the metabolic rate would go down to this level, which is the baseline level here, just like Lee et al. Were doing it. And then, but the metabolic rate remained high and approached the baseline level, but it never really came all the way down there, back to the baseline level. And the biggest problem here was that we didn't have data points here and here and here and here as we were coming, or as the fish were coming down to that level of metabolism. And that makes it really hard to estimate the anaerobic component, because this line could go like this, or it could go like this. And that would, because the recovery is so quick, such differences can make a huge difference in terms of the estimation of the anaerobic component. So we were unable to estimate any epoch for these fish. If we take a look at the pectoral fin use, then here we have the pectoral fin use of these individual 18 fish, and then we see that the steady swimming cost increases as the pectoral fin use in the individual fish increased. So that suggests here, as visualized with these images of rainbow trout, which I know is different from guppies. I've just used them to illustrate the point, that when the fish is swimming without using the pectoral fin, then the swimming costs are low, and when they use or extend the pectoral fins as well, then there's much higher swimming costs, as shown on this graph. And then, and simultaneously, if we correlate the pectoral fin use and then the critical swimming speed, then we see a negative correlation. so that a fish that extend or use the pectoral fins a lot, they have a lower U-crit, in contrast to fish that have very limited use of the pectoral fins. They have quite high critical swimming speed. And then I should mention that these relationships were not explained at all by the maximum metabolic rate. There are certainly variation within the maximum metabolic rate. It was ranging from about 950 up to about 1500 milligrams of oxygen per hour per kilogram. But the variation in the maximum metabolic rate, although it's normally assumed that the critical swimming speed is determined by the maximum capacity or the maximum metabolic rate of the fish, that was not the case here. It was much more influenced by the pectoral fin use. Then, as a consequence of these graphs, if we plot the critical swimming speed against the steady swimming cost, then we see that fish with a high critical swimming speed have quite low swimming cost, whereas a fish with a low critical swimming speed has very high swimming cost. And obviously that is what's driving the metabolic cost up, that this is the high steady swimming cost in the low critical swimming speed, and that means that they meet the maximum metabolic rate and therefore they cannot swim any faster, and therefore that happens at a low swimming speed. If we take a look at the guppy reproductive traits, then we have the wet reproductive allocation, which the range was from about 5% up to about 25%, and the dry reproductive allocation was from about 1% up to 26 or 27%, and the pregnancy stages were ranging from 0 to 50, and in this case 0 is basically the animal is not pregnant, and with 50 then the offspring are ready to be born. So it's basically the full range of pregnancy. Yet we found no relationships with the steady swimming cost, the standard metabolic rate, or the critical swimming speed. Those traits were not explained by the reproductive traits, and I can give you an example here. We have the pregnancy stage here going from 0 up to 50, and we have the critical swimming speed. There is a negative correlation, but it's far from significant, and it explains only 5% of the data. So that suggests that the pregnancy stage in these guppies does not really determine the critical swimming speed, which suggests that these two fish, even if they are quite far apart in the pregnancy stage, they have the same swimming capacity. With that, I'd like to move on to the conclusions. We saw that the transition from pectoral swimming to a combination of pectoral and tail does not reduce the cost of transport, and the transition from steady to unsteady does certainly not reduce the cost of transport. So gait transitions in fish are not associated with a drop in metabolic rate and cost of transport. We have seen that the interindividual variation in anaerobic metabolism can contribute more to the diversity in swimming costs than the interindividual variation in aerobic metabolic rate. We saw that the metabolic rate, the aerobic metabolic rate is only ranging within this small range here, as compared to the range with that includes the anaerobic component. Suggesting that the anaerobic component should be taken into account when interindividual variation in metabolic rates are measured. And then we saw that the guppy, the aerobic metabolic rate continues to increase after the transition to burst-assisted swimming. Here, for example, guppy number 17, when the fish were bursting, then the metabolic rate was higher, which is a surprise because now the animal is It's using and this is the aerobic metabolic rate, so it's not taking the anaerobic component into account, and it might be hypothesized that the aerobic metabolic rate should go down when the fish is also using the white muscle, because why should it start using the white muscle if it still has capacity in the red muscle? But that's what the data shows. And then we have, we found that variation in pectoral fin use is associated with inter-individual diversity in swimming cost and ucrit. We saw that the more the fish are using the pectoral fins, the higher swimming cost they have, and the more they are using the pectoral fins, the lower critical swimming speed they have. And then there are relationships with suggesting that a low critical swimming speed is associated with high swimming cost. And then finally, we found no evidence that reproductive traits are correlated with metabolic rates or swimming performance. With that, I would like to move on to acknowledgements. I'd like to thank Amanda Bennett, Andrews Jordan, Christian Turacci, John Svendsen, Kim Åstrup, and Brune Christensen for their help with all these studies. And we have received funding from Danish Research Council, the Research School SLIP, and the FishNet Research Network, the IDELA Foundation, and National Science Foundation. And with that, I'll say thank you for your attention, and I'll be happy to answer any question that you may have. Yep.
So why weren't, with the guppy, the second one, why weren't you able to, I understand that happened really quickly, but did you just not measure that because it's not feasible to get it that quickly, or because the other species you tested, it took a little bit longer for the decrease to happen?
That's right.
Okay.
It is possible to measure, but it is more challenging to measure at a high rate, simply because you need to have the decrease in metabolic rate within a time span. If we go all the way back to the R0 figures here. Oops, not there. Sorry. That one. Then, if you want to measure it really with a high resolution, then you'll basically be splitting this graph into smaller parts, and your measurements become a bit unreliable, or you really need a steep decline, because if you only have this time, then your fitting of an equation here becomes challenging if you don't have a very steep line with very little noise in. So if there's a little noise and the slope is not very steep, then it can be difficult to measure the oxygen consumption rate at such a high frequency. Yeah.
Thank you, John. This individual variation, I think, is quite interesting. I've looked at this in integrated snails,
All right.
where I found, groups, like groups of 5, you'd always get the same rate. But if you looked at individuals, they varied a lot. But if you looked at the same individuals the next day, they didn't look like they did the day before.
Oh, so they were all changing.
So the question is, would Guppy 2 still look like Guppy 2 if you repeat it?
Right. We did not do any repeatability studies here, but repeatability studies of the standard metabolic rate and the maximum metabolic rates have been done, and they have shown that the standard metabolic rate and the maximum metabolic rates are repeatable at least over up to, I think it's up to 5 weeks or maybe even 10 weeks in fish, measured not with a swimming flume, but with in a resting respirometer, where you measure the standard metabolic rate, and the maximum metabolic rates have been measured by the chasing protocol, where you basically exhaust the fish and put it into your metabolic chamber or the respirometer. And when using that protocol, the measurements are repeatable at least over weeks. But the relationships or the repeatability certainly declines and eventually disappears after, I think, 5 to 10 weeks. So it's not throughout the lifetime necessarily, but at least over weeks. Yep.
Because this is sort of playing into another thing that's hot, you know, on the whole personality concept, that you have fish with bold fish versus shy fish and things like that. And there, you know, the whole spiel about it is, okay, if you want to claim that there are such things like bold and shy fish, they have to be consistent over time. So I find this really, really interesting. I wish I had your presentation or some of the slides for the fish ecology for the fish. Okay, we have like a swimming and metabolic rate kind of a lecture. Perfect stuff for that.
I'd be happy to share this presentation with you.
I would actually like to, you know, like ask you for
That's fine. That's fine.
one of these slides for that. And why do you choose the surf perch? It's so funny because it's like a Pacific coast.
Right. Much of this work was done while I was taking a class at the Friday Harbor Laboratories at the University of Washington. So the surf perch, they were available. They're fairly easy to catch net seining, beach seining.
Yeah.
And also they will swim in the flume. It's always and they will start bursting, and they will do the transition from pectoral fin to a combination of pectoral fin and caudal fin. So in that sense, they are. I would consider them ideal species for that study.
I remember when I was going to the class in the first place, but did you test it? Are these protocols doing this routinely perhaps, that you could make an argument that the eucrit is too low because you're already exhausting somewhat the fish doing these steps? Is this a concern in you kind of? Are you concerned about that, whether you let them, like, you have like 7 steps, and each of these steps is, say, 25 minutes, they say here. So at the end, the fish might already, the eucrit might actually be higher. It's just, you know, course.
There are studies looking into that, and you're absolutely right. The shorter the duration here,
Yeah.
the faster the eucrit.
Oh, yes, that the shorter the duration, the faster the eucrit you get.
Yes, and that is partly because then if your steps are really short, then you can have a lot of anaerobic metabolism going on at the higher steps. But since the steps are so short, then the fish doesn't get exhausted through that step, even if it's relying on at least partly on anaerobic metabolism. And therefore, because the steps are so short, then the accumulation of lactate and depletion of ATP and phosphocreatine, etc., that doesn't stop the fish from swimming immediately, and therefore it can reach a higher speed when the steps are shorter.
Yeah, it's very, very interesting. I really enjoyed the presentation.
Thank you.
Any other questions? All right. Thank you very much, John.
Yeah. I know the one fish was terrified. I felt bad for him. I mean, usually I don't really have like that feeling. The triggerfish, the triggerfish, yeah. And the one where he was just swimming, he was like,
Well,
okay,
he
whatever.
did catch
And then
the
the one where we